Is it going live or is it not yet live? Okay, it seems to be live right now. Okay, very happy that there is a live stream going on. Uh, Thank you. I hope you stick around. Do not conk off. Do not conk off. I am still live. Thank you for staying on live. Yeah. Yeah, Zina, check your email for the live thing link. Can you find it?
No, just let me check again, refresh and all.
Hmm.
No, I can see a meeting link.
What is that? I have
No,
sent
I
you an
cannot.
email. Check your email to Gmail accounts.
Just one second.
Okay, let me even see. Open my sent one.
YouTube.live. Okay, got it.
You can post it in all the three ones, right?
Okay, alright. You are live, I can see you.
Yeah. Okay. So we have some people here. I'll start discussing the internship as uh, the more and more people join in. Thank you very much for joining in. Let us just post it in all the groups. Just give 1 or 2 minutes for this time, right? So there are some more people joining in. Give me 1 sec. Zenith has just posted the live link in the microbiome groups. So people will be, I believe, joining in a minute or two. I can see that in the second group. Uh, Give me a minute. And what Zenith and I did, we were really afraid if the live session may or may not work. So I have also recorded the main message. So in case some of you are not able to join in, or if there is a glitch, which can happen, as you know, that the message goes to you. I'll be discussing all aspects of internship broadly. Just bear with me for 2 minutes. Just want to make sure that as many people are joining in. So most of you would know that this year's Nobel has gone for small RNA. Uh, you'll also be working on that, but not only that, it's a more disease-driven question. Whatever is relevant, you'll be working on that. Just making sure that everyone gets the message before I start. So I apologize for 1 or 2 more minutes of delay. It's 5:04 instead of 5. Give it 2, 3 more minutes so that everyone who has to join can join in. Thanks a lot for your patience. Epilepsy. Okay, guys, we have sufficient number of people who have joined in. Those who haven't, we will be sending them messages of the recording. So let me explain the first principle. We haven't um, put doors um, behind sort of to let people in. As long as you're working in the right domain, life science, or you're from data science AI background, you're interested in research, we will let you in. Now you may wonder, what kind of research are we going to provide? I'll just give you an example, some of the people involved. Brooks Robinson is the program director. He's the head of Lydia Center for Human Resilience. So one of the very well-known brain research institutes in the US. He used to be my junior when I was at Texas and a very good friend collaborator. I actually ended up being his sort of co-PI, and he did 7 different projects with me. So Brooks is one of the mentors. There is Alfredo Gezi from University of Puerto Rico. There are 2 scientists who will be joining some projects in the last phase who are from MIT. There is a lady, Heather Reed, who's very well known in epilepsy from University of Connecticut. She'll be joining. Then there are a bunch of scientists, mostly from India, not exclusively, who have rather recently completed their PhD, who will be guiding the teams and will be conducting research also. It's not just that they'll be guiding. Uh, we'll see how many people eventually remain with us. So So our approach has been to take along as many people who want to come in, provide them two kinds of training, and we do understand the medium which we have chosen, which is WhatsApp, is considered to be social media, is not really seen as a very serious way of education. So not every message may be going through, or you may not be paying attention simply because it's on WhatsApp. Um, we are all humans, I can understand that, but we are putting in information which is very relevant and driven by our philosophy. It's not a generic program. We don't want you guys to be technicians here. We expect you to be scientists. Um, we will not appear as the most friendly people in the beginning simply because we're dealing with an insanely large number of roughly 3,000 people. I do expect by the time we get to research, this 3,000 number will be down to 200 or 300 people who are really, really serious. And this 200 or 300 people will be working with around 60 mentors, a team of 5, 6. If the number of people who go on to the next stage is high, each mentor can have two teams or three teams. But your individual group size will not be more than six, and you'll get individual attention. Each group will have a common denominator of me and Brooks. There'll also be data scientists and AI scientists, which will be taking your work further than just bioinformatics can take. Now, let me address another question. Do you need to already be proficient in bioinformatics to be in this group? The answer is only partly yes. So let's say you haven't done any real bioinformatics work, it was part of your curriculum, or have you taken some videos, we are still delighted to have you. You still have a wonderful chance of making it to the research stage. Obviously, you are coming with a disadvantage that you have to learn more compared to somebody who's already proficient in bioinformatics. Um, so, obviously, having a bioinformatics and computational biology background is a big plus. But even if you don't have it, please don't get afraid. Okay, be calm, learn things. There'll be enough lanes. When I talk of the issue that will be guiding you with our philosophy, since we don't want you to be mere technicians, we'll be discussing two things which no other bachelor's, master's, or PhD program usually discusses. Um, Definitely not internships, because internships are only interested in getting stuff done from people and handing them a certificate, moving on. We will be very strongly discussing how to be a very good science communicator, how to write excellent scientific papers, how to give good conference presentations, how to ask proper questions, how to even write good grants so it helps you 5 years down the lane. We're interested in overall education. So the company which I founded is has people who are like even people who are based in India. Abhijit Banerjee, he used to be the head of biology at Calgary. We are a team of scientists who have worked at highest level. I'm sure you know about my collaborations. Some of us have worked with Nobel laureates. Almost all the top people have worked with NAS members, worked on cutting edge questions. So we are interested in trying to provide you the most quality research training. When it comes to question, you can understand the question is very relevant that we are addressing. I'll get to that. But those of you who cannot go to the next stage, if you're sincere, you try, and after trying you fail, it's okay. We'll still give you a certificate of participation. We'll give you a certificate of appreciation also. And even if you don't fail, but you say, no, no, I don't have time for long research, after 1.5 or so of training, I want to be off, we'll still give you other appreciation, maybe even LORs, depending on your performance. So we want to make sure that we are super responsible towards you if you're sincere. All right. So you're not going to lose out on your effort if you don't make it to the next round. And the next round is not a threshold that I only want 10% people. I wish all of you pan out. But having done this kind of work since I moved back from US to India in 2014, having done large scale citizen science projects, I roughly have an idea that around 10 to 20% of the people who join in actually pan out. So just basing it on my previous experience, that is the number I expect to go till the next stage. But I would be pleasantly surprised and delighted if the percentage is higher. Now let's get to what you get if you do research. Research authorship is in the order of your work, not based on your skin color, nationality, age, gender, affiliation. That's it. Be a better communicator. I've previously run a internship where 9,000 doctors worked with us. Around 1,000 completed their books. We put them online on Amazon and all. We gave them certificates. These were mostly doctors, so they were looking to move to US, USMLE, and there were also some who were in the UK PLAB program. And a lot of them got into the higher thing, and they were very thankful to us for the help and guidance. But there we realized that we faced unusual challenges from some people in Middle East. We had equally good response from Pakistan and India students. That's the first time I worked with Pakistan students. I had few Pakistani friends when I was in Austin, Texas, and one Pakistani friend when I was in New York, but really no working equation. And I realized that there is practically no difference in quality. You get the same spectrum from wonderfully brilliant to good people to absolutely helpless cases. The same spectrum you get across India, similar percentage. So I guess broadly anyway the same people, whether it's Pakistan or India. But we had language issues when we worked with some people from Middle East. So if at all English is not your conversational language, we don't care what you speak at home, whether you speak Arabic, Hebrew, Chinese, Japanese, Hindi, Tamil, Urdu. We don't care what you speak at home. But since the medium of communication for research will be English, if it is your weak point, please try to improve it. This is not coming from any linguistic bias. It's simply the fact that English is lingua franca for research, and we conduct all our business in English. So, if there is a challenge which we had previously encountered, that's why I'm just sort of warning ahead that if you have some issues, and my accent is more understandable to Indians and Arabs, I can switch to more American accent also, having lived there for almost 15 years. But some of the other top people in this project, like Brooks and Alfredo, are from US. So I hope you can follow their accent. Majority can. I've never had this issue, except two or three students. But I'm just sort of raising the issue also. What will be the language of communication? Now let's get to the training. First part is we are posting a lot of links on how to write well. I will soon be posting even more difficult, very advanced level things of YouTube, almost 10 hours worth of videos by different people. TEDx talks, TED talks, some people, University of Chicago top people who actually one professor who's considered to be absolute master in communication. Most scientists ignore them. Most scientists even ignore a scientist like Steven Pinker. Some of you may be aware of a neuroscientist, Steven Pinker. I may not agree with everything he has published, but undoubtedly the guy is a genius in his communication. In his books, he's absolutely amazing. He's one of the most terrific science communicators of our time and is an inspiration. He teaches at Harvard, and I think he has done a lot of research, not just at Harvard, even collaborated with a lot of people from MIT of late. And his work again is becoming very relevant in the age of AI, where we want to understand how not just humans learn or animals learn, but how neural networks can learn better than they're already learning. So I'm even posting some of the videos. Now, for some of you, it may be very advanced, but it'll be a shame if you ignore it. If you actually learn from it, it'll be really, really good. But we will, in few days, go to the basics. The basics will include the standard thing of a structure of a research paper, structure of a review, what are the standard reference formats, right? What is a Chicago style formatting for references and all such basic things that you consider. Um, how ethical is it to use AI for part of the writing? What are the challenges? Forget even ethics. Even what are the problems? All right. If you how to actually use AI for better literature search. Now, some of you may not be interested. So, initial module will not teach you Python programming, but those of you who continue with us beyond the initial training, we will try to also teach you data science and AI related to the project. We will try to teach you some structural computational biology, predicting structures, doing docking. We are interested in sort of drug repurposing. We'll try to teach you some RNA computational biology. We'll try to teach you metabolomics. We will also try to teach you some even GWAS studies because we like to look at population and all, see how they respond because there is not just microbial data, there's also data on individuals who live to a century, and we like to look at the correlates, but that's very advanced stages. We're not talking immediate steps. So the immediate paper, let me address the sort of big question of what we're going to do immediately in terms of our paper. The question is very simple. The first step, I'll go into all the steps step by step, but first step is a lot of bacteria can synthesize serotonin. Serotonin, higher amounts roughly equal to less seizures. Higher amount of serotonin roughly equals less depression. Higher amounts of serotonin is also good for lesser neurodegeneration. Obviously, I'm talking in very broad strokes. There are nuances. In fact, my own experimental work with Nace Golding at UT Austin showed this is not something I published and sort of planning to, as I'm moving back to US probably next year or next to next year, planning to address it experimentally again. That is that serotonin is not just paracrine. It can be both paracrine, autocrine, yet synaptic, and can be depolarizing yet inhibitory. So we found a lot of interesting things, and we found it to not just work alone, but work with norepinephrine. So I basically, whatever I told you a step before, I found a lot of exceptions to it, and that's the beauty of biology. Whatever you find as a rule, there can be exceptions depending on the brain region. So now, stepping back from the aside, broadly, if the serotonin is up, you have fewer seizures and you have fewer other neurodegenerative disorders and you have fewer depression problems in very, very broad strokes. So, what we would like to do is understand which bacteria can modify, which bacteria can sequester, which bacteria can transport, which bacteria can break down, and which one can synthesize serotonin. The first approach to it is very simple. We already know from others' work. We are standing on the shoulders of giants. From others' work, we know the genes which are involved in serotonin synthesis, not just in eukaryotes, not just in humans, but in bacteria. You can take the sequence, you can find the nearest bacteria, not just straight away go to the final one of your interest. Let's say, hypothetically, let's say you do not know. I know this is a wrong example. We actually know in Bifidobacterium. But let's say you know about the gene involved in E. coli and you were to figure out for Bifidobacterium. You can run the gene, tryptophan hydroxylase or some other gene, just using it as a dummy example, and look for sequence homology in Bifidobacterium. There could also be a better approach where you look at portions of genes and you look for the closest bacteria, then you look for something intermediate to Bifidobacterium. So if the evolutionary distance is high, which can be a very high among different bacteria, you're still able to find the gene. You can even validate it by looking at its structure and looking at is it interacting with similar partners. So it's not just the gene, but the gene cluster involved in the final serotonin synthesis. Are all of them there, right? So that's the first step. Now, within the first step, you'll actually be learning some very important technical aspects. Is BLAST sufficient or is it wrong? Because BLAST looks at overall homology, right? It's looking at global minima, which can be wrong at times, but it's very fast approach. If you were to do overall sequence matching, that's more accurate, but it can be extremely slow. So how do you solve this problem? So we'll go over those technical aspects. Now, for those who are just undergoing training, may not move to the next stage, you will learn simply how to write well, how to communicate well, and everything related to search. The first step I told you. Beyond that, the training ends for you. You don't clear it. You move on as long as you appear, and there's a sincere effort. Those of you who clear this step, you practice with a dummy data set. We are sure that you can do this well. Then you get assigned some bacteria, and that's your first step. But then the questions that follow are rather interesting. The questions which follow, I mean, this itself is interesting, but the questions that follow are rather interesting. How can some common molecules from diet affect it? How can common medicines that we take affect it? And obviously, you can understand how to solve that question computationally. It's not very difficult because you will take uh, the structure of the molecules involved in direct synthesis and you look at common medicines and you'll try to do docking and see if the docking works. If docking works, you'll do molecular dynamic simulation. You'll also be looking at signaling pathway. Then you may look at metabolite regulation. You may also look at RNA regulation. And whether you do it in the same paper or you do it more may not just depend on the technical aspects, may also depend on your hurry. Let's say some of you, after the training, only have 3 months left, and your whole team is organized in a way that you would like to see the paper come fast, not necessarily to the topmost journal. We may try to accommodate you depending on your performance, right? You must understand, if you're performing really well, we'll listen to you, you add value, and if you're not performing well, we may not care because we have hundreds of people to handle, right? You must understand that fair thing. But if we can accommodate you and you would rather like to see a smaller sized publication, we may just stop at the first question. And we'll also see, we don't want to do minimum publishable unit, but we also don't want to stuff a paper. One paper, one message. Now look at how big the scope is. We want to go through every single microbial species in the microbiome and address it. And that is why this large citizen science effort has been sort of unveiled. You can understand how logistically challenging it is for us to initially manage thousands of people and then manage hundreds of people. But if we can look through every single bacteria and address the direct synthesis question or direct, breakdown question, so direct contribution to serotonin amount. Then will be the indirect, which might actually be bigger. Then will be the question on specifically effect of diet. Then will be question of repurposing existing medicines. Then there'll be question of effect of exercise. There'll be specific keto diet. Is keto diet just working because of keto bodies, or can it work because of something very interesting, such as altering serotonin? I mean, that'll be groundbreaking, not just important work. That'll be groundbreaking work. And we currently as IonCure don't have much experimental facilities. Whenever we publish experimental work, that has been in collaboration with some labs in Austin, Texas, with my friends. And in fact, one of the top friends lab is currently not working. So we will be seeking out some new collaborations for the experimental part. But by and large, the collaborations are basically bioinformatics ones. Already there are very accomplished people who have joined this group. There might even be more. Um, some top people from experimental labs. I'll look into that possibility around December time. I don't want to look ahead of time because if I tell them I have 1,000 people, but let's say only 50 turn out to be good, I don't want to give them a wrong estimate. I do have some very good friends, one or two originally from India, mostly Americans born in US, who are running microbiome labs in US. Now, let me even address the last point before I close down. I think I've tried to explain to you everything. There's another shorter video which we'll upload, and we will even create a form, because after this video, I like to ask everyone, what are your questions? See if the question is already there. If your question is not answered, Arsh will create a Google form that you can send a question. We'll keep updating the form so that if your question is already there, you don't repeat it, right? Last question is people who really work with us and have been successful. When I was a professor, after coming to India, I was professor at two, three places. I will not go into it, rather bad experiences in India, and compared to the top quality of US, Indian research leaves a lot to be desired. But I had 27 people of students who worked with me and who got PR in Canada. So people, if you Google my name, I think a lot of you have already seen that in India I've given maximum number of TEDx talks. People who come to me come because I do a lot of citizen science and I've helped people to find the right places. Okay, we don't pay, we don't charge. I mean, a lot of people even ask, why don't you charge people? And we just feel that at least so far we haven't done anything that spectacular. I mean, if we do something in even more organized way, we may start charging. But at least for your batch, we are not charging anything. We are not paying anything. The kind of people who usually come to me and are super happy are people who are looking to either study or even immigrate to other countries. Like I said, 27 people moved to Canada on PR for the 3 years I was at CDRI. At ICGB, I did not train much students. I was actually took a temporary position under the director, and I worked with four people long distance. But at CDRI, there was a mass thing. There were thousands of people, even long distance. So we have done that, and as a company, we have been very helpful, I and Cure, to people who have moved to US for studies or moved to Canada, Australia, New Zealand, Ireland for both studies and immigration. So people who are looking to really improve their profile, work with top American authors, they are most suited. If you do some small thing, you don't clear the next thing, we'll still give you a certificate. It doesn't do any harm to us by issuing you a certificate, as long as you're not trying to lie to us. Once in a while, we do get people who try to tell me a lie, thinking that since hundreds of thousands of people are working, we'll not have record. They'll try to say, I did this, I did that. I ask them for report. I say, you made it now. As long as you're not doing it, we don't mind giving you certificate if you do the bare minimum threshold of answers in how to write a scientific paper and gene alignment. But frankly, those who will do well with us will move on for 6 months, 8 months, or even a year of research and earn some big publication. There, what we will do is we'll happily write LORs. My American colleagues will happily write LORs. We'll give you certificates. We'll recommend you on LinkedIn also. So basically, it's a career boost, whether it's for immigration or studies abroad. That's where it's a perfect fit. So you need to ask yourself whether you have 20 hours, whether you can put up with us for a month and a half, now less than month and a half, of messaging through WhatsApp, because I realize WhatsApp is not the most uh, perfect channel. In fact, we thought we'll do 1.5 months only on WhatsApp. I am reconsidering it based on some feedback. Uh, but let me see. I'm not making any promises there. Those of you who are active will try to read everything we are posting on the WhatsApp groups, try to go through every single link. We will see if we want to onboard people, 1,000 plus people, because it's currently 2,800 or something in the three WhatsApp group. If you want to onboard that many people on Slack, challenge is just the logistics of it. So we are trying to keep the initial phase simple. We may even create a Telegram group. Telegram, the nice thing is the information is not stored on your device, it's stored in that, so in the cloud. So it's somewhat better for you in terms of handling things. We may add few more channels of information for the initial phase. Some of you who are very interested in starting bit sooner with dummy data set, we will also do this. So right now you'll get three kinds of information. What is the question, right? Mixed with how to write well, mixed with, after sometimes staggered, how to do gene alignment well. So this is the training that is there planned for 1.5 months. And mostly through WhatsApp, likely through Telegram. We'll see if you want to do some more thing. We have your email IDs if you want to do some even email lists. We'll go through such things. We want to make sure you get all the information. Next time soon, in a day or two, likely even tomorrow, we'll have a Q&A session. We'll find some other platform where we can have Q&A, because right now only 242 people have joined. So if 240, 250 people join, we can work through other platforms, need not be YouTube live. I'd like to end this meeting and be very thankful to all of you who have joined this. Um, Please be assured there will be forms on the frequently asked questions so that your queries reach us. We just don't want the WhatsApp to be flooded with thousands of messages. That's why we have put it as only admin only messaging right now. Thanks a lot for being here. I hope I have answered some of your questions. There will be some more messages soon, and I hope it's going to be great learning experience for everyone who's on board. Thank you. Bye bye.